Welcome to this lecture on sequence file formats in computational biology. Think about how you save a text document on your computer. You might use a simple text file, a Word document, or a PDF. Each format stores the exact same words, but organizes the underlying data differently behind the scenes. Today, we will explore how we do this for biological data. Building on that idea of saving documents, in computational biology, we deal with DNA, RNA, and protein sequences. These are essentially massive strings of alphabetical letters, representing the fundamental building blocks of life. We need to save these biological letters in computers so that sophisticated software programs can read, analyze, and compare them. If every scientist around the world invented their own personal way to save these sequences, no computer program could read them all. Global collaboration, which is the absolute cornerstone of modern scientific discovery, would be completely impossible without a unified approach. To solve this problem of collaboration, the scientific community created standardized sequence file formats. A sequence file format is a strictly standardized, text-based method for representing and storing biological sequences in a computer file. The term standardized means there are rigid, unbreakable rules about exactly where the sequence starts, where the name goes, and how extra information is written. By adhering to these strict structural rules, these formats allow bioinformatics tools and global databases to seamlessly exchange and parse biological data without any human intervention. Now that we know there are strict rules governing the sequence data, let us look at what else is stored inside these files. These files also contain metadata, which refers to crucial background information about the sequence itself. This can include the specific organism it came from, the scientist who originally discovered it, and the known function of that specific gene. When a computer program opens one of these files, it uses the formatting rules to automatically separate the biological sequence from these human-readable notes. This automation is the absolute foundation of all modern computational biology. This automatic separation brings us to why these specific file formats matter so much in real-world research. Imagine you are a researcher downloading 1,000,000 DNA sequences to find a genetic mutation linked to a rare disease. If the computer does not know exactly which line of text contains the DNA and which line contains the patient identifier, your analysis software will immediately crash. Standardized formats like FASTA are used universally by powerful alignment tools, such as BLAST, to rapidly compare sequences across different species. Meanwhile, richer formats like GenBank are used to store heavily annotated information in global public databases. Before we look at the specific structures of these files, it is highly beneficial to understand the mathematics behind sequence data storage. Biological databases contain billions of sequences, so we must calculate how much digital space these files will consume. We can model the total file size using a linear algebraic equation. S equals H plus L times B. Here, S represents the total storage size of the file in bytes. H represents the size of the header, or metadata. L represents the length of the biological sequence, and B represents the bytes required to store a single character. As the sequence length increases, the total file size increases linearly, but formats with heavy metadata will have a much larger constant H, taking up significantly more space. Now that we have our storage formula, let us apply it to the FASTA format. Suppose a researcher saves a newly discovered viral DNA sequence. The header line contains exactly 30 characters, so H is 30. The sequence itself is 5,000 base pairs long, so L is 5,000. Assuming each character takes exactly 1 byte of storage, B is 1. We substitute these values into our equation, multiplying 5,000 by 1 and adding 30. The final calculated value is 5,030 bytes. This tiny metadata overhead makes FASTA highly efficient for storing millions of sequences. Beyond just calculating the physical file size on a hard drive, we can also measure the actual biological complexity stored inside that file using principles from information theory. The formula for this is I equals L times the base 2 logarithm of A. The variable I represents the total theoretical information content, measured in bits. L is once again the length of the sequence in characters. The variable A represents the size of the biological alphabet, which is exactly 4 for DNA sequences and 20 for protein sequences. When the alphabet size A increases, the logarithm evaluates to a larger number. This mathematically proves that protein sequences pack significantly more biological information per letter than DNA sequences do. Let us see this information formula in action with a GenBank file containing a functional human protein sequence. The sequence is 600 amino acids long, so L is 600. Because it is a protein, there are 20 standard amino acids, making our alphabet size A equal to 20. We substitute these into our logarithmic equation. The base 2 logarithm of 20 is approximately 4.3219, which is the number of bits per amino acid. Multiplying this by 600 gives us approximately 2,593.14 bits of pure biological complexity hidden inside the sequence itself. Having calculated the storage and information metrics, let us visually compare these two formats. On the left, you see the FASTA file structure. Notice how simple it is. Just a single header line, starting with a greater than symbol, immediately followed by the raw sequence data. This is built for pure efficiency. On the right, you see the GenBank file structure. It is much more complex, featuring distinct sections like locus, definition, and features before we even reach the origin where the sequence begins. This structure is designed to hold rich biological annotations. This visual comparison highlights why we need different formats for different tasks. The FASTA format provides a lightweight, universally accepted method to store raw sequences using a simple header. This makes it absolutely perfect for rapid computational alignment, where speed is critical. In contrast, formats like GenBank provide rich, heavily structured environments for storing complex biological annotations alongside the sequence data. Both formats form the essential digital infrastructure that makes modern computational biology possible. To wrap up our lecture, let us review the key takeaways. First, standardized sequence formats are what allow bioinformation tools to seamlessly exchange and parse biological data without crashing. Second, we learned that total file size increases linearly with sequence length, but heavy metadata adds significant overhead. Third, the true biological information content depends on both the sequence length and the size of the biological alphabet. By understanding these structural rules and mathematical principles, you are now fully equipped to handle real-world genomic data.